Okay, good morning. Today we'll continue our discussion about how eukaryotic cells, like the cells in our body, have different opportunities to regulate what genes that they are carrying around in their genome actually get used in order to determine certain cell type specific functions. And we'll also kind of revisit this idea of what determines cells differentiating, starting from stem cells that have the ability to become either a handful or potentially any type of cell in our body. How is that process of cells determining their fate actually a function of different gene regulation decisions during development? So let's start where we left off last time. We ran out of time last time and didn't get to this idea about stem cells really becoming differentiated cells. We did talk about this slide, and I think it's important that we start off this lecture kind of reviewing what this was about, because this is a really important concept, right? I said that your body is made up of roughly 220 different types of human cells, and those 220 types make up the 30 plus trillion cells that make up your body, right? So as an example, we said, what if we had three different differentiated cell types, right? Differentiated means these are cells that have become the final form that they're meant to be, whether we're talking about in this picture here, a skin cell or a nerve cell or a red blood cell. Very different types of cells with very different functions, right, that do different types of jobs in your body. But yet we all know, and I think you know, of course, that all of these originally came from stem cells, right? Stem cells that give rise to things like your skin, your nerves, and your blood components. And if you go back in time enough, all of those cells originally came from the one fertilized egg, right? That was you 20-something years ago. Yeah. So if we think about these cells in our body today, as you're sitting there watching this lecture, right, inside of these cells, each one of them has the same master blueprint, your genome, right, that you got from mom and dad. It's really just a matter of those, roughly 20 to 25,000 genes that are contained in the human genome. What subset of those genes are these particular cells using in order to be a skin cell, nerve cell, or blood cell? So we said that let's imagine that you looked at just one gene somewhere, right? You pick a chromosome and there's one particular gene. And so all three of these cells, of course, have this present inside their nucleus, but these different cells may not be using this gene the same way. We said that maybe skin cells, in this case here, maybe they have a sort of low level, but sort of constitutive or constant level of gene expression of this gene. Right? So here, for example, the color coding on the gene, this is just a strand of your DNA, right? It's some chromosome somewhere. And we said maybe this is the coding region or the actual transcribed region of the gene in blue here. This green area would be the promoter region, right? And then you'd have some of these other elements that help regulate them. So remember, our analogy was if this is an airplane at the airport, right, the green part would be the cockpit of the airplane, where you have the pilot, co-pilot, that ultimately has the most intimate control over what that airplane does. So in this case, the promoter is where general transcription factors, these sort of generic purple proteins, right, they pile up on the promoter, and that forms the right three-dimensional shape, the right landscape of them all piled together to recruit RNA polymerase. And so RNA Pol2 begins transcription there. We said other regulatory elements that might be close by or really might even be very far away are actually controlling the degree to which the RNA Pol2 is functioning from the promoter, right? So this would be the control tower of the airport radioing the pilot and co-pilot to tell them what to do. So we said in this cell here, maybe there's just a regular low-level constitutive level of expression. So you have RNA Pol2, transcribing this gene, and then ultimately what the RNA that's made from that gets processed into mRNA, and then the mRNA is translated by the ribosomes in the cytoplasm to make a moderate level of this protein, right? We said, what about in nerve cells? What if in nerve cells, in order to be a nerve cell, it needs to have a lot of this protein made, right? In that case, hey, it's the same gene, right? It's the same genome that it's working with, but here you have some sort of other transcription factor protein. In this case, the nerve cells have this yellow protein, which is a regulatory TF or transcription factor, and it can bind to an activator region, right? Maybe like an enhancer, for example, right? And that greatly increases the activity of the RNA Pol2. So the nerve cells make a lot of this protein compared to, let's say, the skin cells, right? The other example was maybe red blood cells as they mature and become red blood cells. They don't need to make this protein at all. Whatever this protein is totally irrelevant to being a red blood cell, right? So as a result, that yellow transcription factor is not present in these developing red blood cells. And instead, these developing red blood cells have a different, so this red diamond transcription factor instead. And that one binds to another regulatory site that talks to this gene, but in this case, it's a repressor site, right? So it ends up inhibiting RNA Pol2 from starting transcription at the promoter, and as a result, the developing red blood cells don't make this protein at all. So what I asked you to think about at the end of last lecture was, what if this protein that's being made here, this blue pentagon, is itself a transcription factor that will then go on to bind to different regulatory sites around genes that it has an influence over, right? So there'd be a whole set of genes in the genome that might have a regulatory site where this blue pentagon protein binds to, and therefore you'd have a wave of genes that are influenced, right, by this, that would be influenced very differently in skin cells versus nerve cells versus red blood cells. In other words, you could look at this cartoon and say, this cartoon then repeats again, right? But instead of this yellow transcription factor being the thing that's different, you know, between the nerve cell, the skin cell, and red cell, now you would say, oh, there's like a blue pentagon, right, as one of these things. And so therefore, the next wave of gene transcription influenced by that would send the nerve cell to be even more different than the red blood cell or the skin cell, right? The other way to think about it is to go backwards in time, right? Because after all, this yellow transcription factor or this red diamond transcription factor, right, itself is there or not there due to prior waves of gene expression controlling those genes that code for the yellow transcription factor or the red transcription factor, right? So if you go backwards in this cartoon, that's how you eventually get back, right, to the starting stem cell, the pluripotent stem cell, which was the fertilized egg that had all of its genes in the genome, right, like a blank slate, right? No decisions had been made about what genes to turn on and turn off yet. But as that fertilized egg began to divide, right, decisions started happening, right? And different waves of genes began to turn off or be turned on, right? That then ultimately led to the creation of the 220 different types of cells that make up the trillions upon cells in your body. All right. So let's explore this a little bit more. This idea that as stem cells differentiate, right, and become more like the finalized, finished versions of all these different cells that make up your body, we are talking about waves of decisions being made that influence waves of genes being expressed, right, and the products of those genes then being the basis of the next wave of decisions. So one analogy that I am fond of is thinking about this like folding origami paper animals, right? I think you're all familiar with origami. You know, a lot of people are really talented. They can make really complicated, wonderful three-dimensional shapes, starting from just a blank piece of paper, right? So let's think about this. When you start off with the same square piece of paper, that is like a pluripotent cell, right? You haven't made any decisions yet about which way to fold it, so everything is just blank, right, and ready to go. Once you start making the first critical fold or the second critical fold, you are starting down a pathway of decision making that will ultimately, with each additional fold you make, limit a little bit more, right, your possible outcomes of what this shape is that you're going to make, right? So you can think about that like a pluripotent cell. When a pluripotent stem cell, like the fertilized egg, first starts making decisions about gene regulation, these are the first few critical folds, right? But maybe in reality, these first few critical folds are sort of very generic, right? With every fold, there's still practically, uh, an almost limitless number of final kind of outcomes that you're getting towards, right? But at a certain point, once you've made enough folds, you really begin to sense that you're committing yourself, right? So here, at this point, the stem cell, let's say you had 3 different pieces of paper and you were all following the same set of early decisions folding, right? So you get to this particular structure here, and so you have a blue piece of paper that you got there, an orange piece of paper that you got there, and the green one as well that's shown here, right? The next fold that you make from this, right, will be the first time that these 3 pieces of paper begin to diverge, right? Because each of these, the next fold is different, right? And from that point on, you are really beginning to commit to finally arriving with additional decisions, additional folds, down to making 3 very different animals at the end from this origami, like a shark, a camel, and a honu, right, a sea turtle. So if you think about this, in our cells, of course, we're not talking about physically folding the cells. Here, the folding of the paper represents, right, it's an analogy for waves of decisions about what sets of genes are going to get turned on now, what sets of genes are sort of not turned on or ignored, right? And with each successive wave of genes that are turned on, that will dictate what the next possible set of decisions can be, so on and so forth. That's why in your adult body today, you no longer have any pluripotent cells, right? The stem cells that you have in your body are all sort of tissue specific stem cells. In other words, they've gone far enough down this decision making so that they are now called multipotent cells. For example, the stem cells that are in your bone marrow that give rise to your different blood cells, right? They can still become, for example, any one of several different types of blood cells, but they cannot become something like a nerve cell or a muscle cell, or some kind of gland cell, right? Because those would have branched off of decisions earlier on in the differentiation process. Okay. So why is this exciting? Because remember, this reminds us of the concept of genomic equivalence, right? Whether or not you're talking about the paper shark, the paper camel, or the paper sea turtle, right? All of them came from the same set of decisions that at some point diverged, right? All of the different cells in your body, skin cell, nerve cell, red blood cell, all still have the history of all those genes, right, as part of their genome. So just like you could unfold the turtle and carefully reverse these folds to get back to this particular shape, right? And then potentially starting from this shape, start folding it again, but instead of folding it towards the turtle, now you fold it towards the camel. So even though you started with a turtle, you backed it up and then went forward again and ended up with a camel. The promise of stem cell technology, the biotechnology of stem cell research, is to be able to do this with human cells, right? To take something like a skin cell, reverse it, turn it into a multipotent cell, or even farther back, right? Further back. The more further back to pluripotency you get, the more options you have for driving it forward to any different type of cell that you need. The example I gave you was the patient with a spinal cord injury, right? How wonderful it would be to take a skin cell from her arm, reverse it to be a stem cell, put it back into her spine, and then encourage it to go forward and turn into new nerve cells, right? And that her paralysis, her paraplegic or quadriplegic condition might be reversed, right? So that's what I wanted you to think about, because I think it's a very sort of heady concept, but it's a fascinating concept, right? Because it really makes you think about, what am I? What am I really made of, right? And this whole idea that you've known for many years that you came from one fertilized cell, right? But did you ever really stop and critically think what has happened to that cell over the last 20-something years, resulting in you sitting here right now thinking about that cell that you came from, right? Okay, let's move on. So we've been talking about different ways or different levels at which a cell has the opportunity to influence whether a gene gets expressed. So we've already talked about the most fundamental level. If you sort of alter or modify the physical organization of the DNA itself to make it less accessible, more packed up in tight heterochromatin, so the genetic sequence is not available for proteins to see it, right? Versus if you unravel it, get it off of the nucleosomes, and then other proteins like transcription factors can then see the genetic sequences, the little sequences that they recognize, right? Then you might be able to encourage genes to be expressed. We moved on and said, well, then it's up to those transcription factors, right? Those different proteins to recognize the specific sequences, small little sequences, that these transcription factor proteins are the right shape to bind to. And that's what starts them piling up on the chromatin, right? Ultimately forming different three-dimensional surfaces that other proteins can come and bind to, with RNA Pol2 finally coming and being attracted to be stuck there and start transcribing. And so that's why, this whole discussion about the importance of transcription factor proteins, right? And wave after wave of, depending on what transcription factors you have in a cell, that determines what the cell is, right? And what it can do. So let's move on. So all of that discussion leads up to the RNA Pol2 successfully docking at the promoter and beginning to transcribe the gene, right? So ultimately you end up, or you start off with, this primary pre-mRNA transcript, right? The primary RNA transcript that comes out of the RNA Pol2. And we've already learned that that RNA transcript, it's protected by a 5' cap, a polyadenylation tail, so that your cell knows this isn't some strange random piece of genetic material floating around in it, right? Because it could be, like viral genetic sequence, right? Like if your cell is infected by a virus, it would have free floating like RNA in it. And so we evolved to kind of want to destroy free floating RNA, right? So in this case, this RNA, our cell made on purpose, right? Because it's part of expressing this gene. And so therefore we have to protect it from being targeted for degradation. So you have 5' cap, polyadenylation tail. And we learned that the actual perfect photocopy of the original gene, right, which is the primary transcript in RNA form, might often contain different parts that end up not really being part of the finished sequence that needs to be translated. So things like intronic regions will be edited out, right? And then the remaining exon regions will be spliced together to make the final edited mRNA that then gets transported out of the nucleus into the cytoplasm, where it meets up with ribosomes, right? And then the ribosomes, as we learned, are these big factories that will then read the mRNA 3 bases at a time, right? The triplet codon sequence. And with the help of tRNAs bringing in amino acids, the ribosomes will chain together those amino acids in the correct sequence that was coded for by the original gene to make a finished polypeptide chain that then folds up as your protein, right? So today we're going to look at the fact that even at this RNA level, this represents opportunities for the cell to potentially interfere with this RNA actually being translated into making a final protein, right? So another level of control over whether proteins finally get made from a given gene. So let's look at that. Let's start by taking a closer look at what is happening as the RNA Pol2 is transcribing the gene. So we've seen this cartoon before. So here's a generic kind of gene. This dark blue region might be the part that gets transcribed. The little black arrow here is sort of the standard notation that says this is the spot that transcription begins, right? So this is where the RNA Pol2, the big purple blob in the back, is positioned, and it was recruited there by all these little transcription factor proteins, right? Like these over here, or this yellow one, this blue one, this orange one, all piling up because they recognize certain sequences due to their shape. And those sequences are found in the DNA, right? Evolutionarily, like they're positioned, these sequences, so that when these proteins are brought there, the proteins themselves form a big three-dimensional shape that is the right shape to attract RNA Pol2. So let's look at this, right? So one of the important sequences that we studied right at the core promoter region is this TATA box, right? Which really literally is just, you will find in the majority of genes in the promoter region, a TATA somewhere very close to where transcription starts. So the transcription factor that recognizes that and is recruited there is called TATA binding protein, or TBP. Right? And it directly binds to the DNA bases. So if you were to look at, several thousand TATA binding protein binding sites throughout the genome on different genes, and you do that exercise where you basically, it's almost like a vote, right? Every position in the DNA in those sites that you're looking at, you look at what base is actually used at that site, right? You can build this type of consensus sequence. And you can see that's how scientists realize it's TATA are the critical ones, right? Followed by, in most cases, a couple of other A's, right? So there are transcription factors like TBP that bind directly to the DNA. Other transcription factors like TF2A or 2B or 2F, they also play an important role in controlling what ultimately happens at the promoter, but they don't necessarily bind to DNA directly. They bind to the other transcription factors that are already there, right? But together, right, the big group of complex of proteins that are piling up there is the right shape for RNA Pol2. So here is RNA Pol2, right? So this is an example of a eukaryotic RNA Pol2 complex. So not surprisingly, it is not just one protein, but it is what we call a holoenzyme, a whole group of separate proteins that are all coded for by separate genes, right, coming together to form a complicated machine, much like how we described the ribosome, right, as being different subunits that come together to form a big ribosome, along with ribosomal RNAs helping it hold together. Here, the RNA Pol2 is made up of 12 different protein subunits. Not all of them are shown in this illustration here, because I think this illustration kind of has it cut in half, so you can kind of peer inside and see what's going on. So a couple of key things I wanted to point out to you, right? One is, if you look in the middle, you can see the spaces here, right? And you can already see that this is where the DNA helix will fit perfectly through the middle of this protein complex. And how wonderful it is when you think that that shape, right, that complicated shape is actually a composite of different contributions of these different pink, purple, etc. proteins, right, that they all lend because they are the right shape and that they fit together to create the space in there for the DNA to go through. The other thing I wanted you to notice is that some of the regions of these proteins actually stick out of this big RNA Pol2 complex. So these are the numbers. I think this is actually yeast, a model of yeast RNA Pol2. but it's pretty similar to what we have inside of humans. I think the numbering is very similar. So you can see some of these RBP protein subunits, that's what they're called, right? These RPB protein subunits. What was I going to say? They actually stick out quite a bit, right? And these parts that stick out are targets for post-translational modifications of these proteins by doing things like adding different small groups, like phosphate groups. So that's why when you see the cartoon up here, the RNA Pol II is drawn with this funny purple tail sticking out of it, right? Now, in reality, it's not going to look like, this kind of looks like a mouse or something with a tail, right? It doesn't really look like that. But for conceptually, right, you can draw it this way to highlight the fact that this tail sticking out is an important target for phosphorylation, when in reality, like the tail sticking out would be just sort of regions of the protein like this. Right? Here you see what they've done is this whole RNA Pol II complex, they've now turned it gray to put it into the background, right? And you can see how the DNA nicely goes through it, right? And this way they can, in brighter colors here, show you how those different transcription factors are all playing a role in getting the RNA Pol II complex stabilized onto the DNA. For example, here's the TBP, right? The TATA binding protein that would bind to the TATA box, right? And here you can see it's playing it's contributing to helping get the RNA Pol II positioned properly at this site. All right. So once this happens, the RNA Pol II will start transcribing the DNA, and you'll start getting an RNA transcript. So why did I talk about that tail of the RNA Pol II and the phosphorylation of that? Because the phosphorylation of that tail that sticks out of the RNA Pol II complex, so by the way, we call it the CTD, or the C-terminal domain of the RNA Pol II complex, right? This is how the cell controls not only the function of the RNA Pol II machine, but also controls what's being done to the primary RNA transcript that's going to come out of this RNA Pol II as transcription is happening. How does this work? Well, the amino acid sequence of this C-terminal tail, if you were to zoom into it and look, you'd realize it's a whole bunch of repeated amino acid motifs. So in particular, there's a motif, a repeat, that consists of 7 amino acids: tyrosine, serine, proline, threonine, serine, proline, serine. Okay, those 7 repeated over and over and over. These serines in pink here can be the site of phosphate groups being added to them. So depending upon what combination of serines have phosphate groups added to this C-terminal tail, this chain of amino acids, the specific pattern of phosphate groups will alter the shape of this C-terminal tail, right? Because depending on which serines have big, bulky phosphate groups hanging on, the way the tail kind of curves around or is folded up would be slightly different. And anytime you're talking about differences in protein folding shape, there is the potential then for that shape to result in other proteins being preferentially recruited, right? Or if the shape is different, right, other proteins are recruited. So here you can see, as transcription starts happening, right, this little red thing in here is the brand new tiny little RNA, the primary RNA that's being put together by the RNA Pol II complex as it's moving down this gene. So as this RNA is going to start emerging out of the RNA Pol II, right away, you want to make sure this RNA is protected from being degraded. Right? So how do we do that? Remember, our cells put a 5' cap on the RNA. So that means the proteins that take care of putting the 5' cap onto the RNA, they better be there as this is happening, right? So they are there because the phosphorylation pattern on particular serines in the C-terminal tail, right, the tail that sticks out of the RNA Pol II, attract capping enzymes, this orange protein. Right? So as the RNA begins to come out, right, this capping protein is already there at the right spot to be able to go to work, putting a 5' cap on that RNA. As the RNA Pol II complex is moving along and the RNA that's coming out of it is getting longer and longer, then the cell actually starts getting ready to do the RNA editing. Right? It's going to splice out introns, keep exons, put it together. And so the pattern of phosphorylated serines on the C-terminal tail changes. So now you attract this blue protein here, which is just generically some component of the splicing machinery. So as you move on to the third thing here, right now the RNA that's coming out of the RNA Pol II, oh, it's getting longer and longer, and now it has a cap. It has a 5' cap on the end. It's not shown here, but there is. And the splicing machinery is ready and waiting for it so it can start going to work. And then eventually, when you reach the end of transcription and the end of this red mRNA comes out, right, you're going to have to put polyadenylation signals on it. So once again, the C-terminal tail of the RNA Pol II complex, right, the phosphorylation pattern on the serines changes, so it now attracts the proteins that will take care of putting the poly A signal at the end, right? So these events that are coordinated in this manner result in the RNA coming out, and it's going to have a 5' cap. It's going to have a poly A tail on it, so it's protected from being degraded. And not shown in this, right, there's going to be lots of proteins on it that are already going to work, editing this primary RNA transcript, right? I mean, come on, isn't that amazing when you think that all of this is happening just because of these dumb proteins, right, that are just doing what they do because things are being done to them, changing their shape, right, and interacting with each other to actually be able to accomplish something as remarkable as this. Right? So let's look at the splicing. So we said as the RNA is coming out of the RNA Pol II, the splicing process begins at the same time. Right? These splicing factors go to work and bind to it. So if you were to look at the pre-mRNA or the primary transcript, you would find on it there are certain regions where you have exons, like exon 1, and then there's an intron region, and then there's exon 2. Right? So these splicing factors that are attracted to the RNA Pol II at the right time, they begin to bind to different signals that define whether there are introns and exons. For the majority of intronic regions that might get edited out, towards the 5' end of the intron, you'll typically find a G and a U in there, all right? Now, this is not to say that throughout the whole intron or whole exon, you're only going to find like this one example of a GU, right? Because let's face it, there are going to be lots of instances where part of the genetic sequence, you have a G followed by a U, right? But what they're saying is having a GU near the front is sort of like a marker where certain proteins recognize and can bind to this part of the intron. At the very end of the intron, similarly, you would tend to find an A and a G towards the end. And somewhere close to the end, there's usually an A somewhere that represents another place where proteins can bind to. So when these proteins bind there, like this pink egg-shaped one and this blue egg-shaped one that are part of the splicing machinery, they come together and they form a lariat or a loop in the intron. Right? And then other splicing proteins will come and remove that loop and splice it out. You'll then have these exon junction proteins, these green ones, that help reattach the remaining 2 exons together. And these exon junction proteins also serve as a signal to start transporting the edited RNA out of the nucleus into the cytoplasm so that now ribosomes have a chance to translate the finished mRNA message. Right? So because of this splicing, we already learned that this alternative splicing and editing of an RNA transcript is what allows our cells to make different variations of proteins all from the same gene, right? Depending on what you edit out and what you keep and what actually makes it into the final mRNA will result in different versions of proteins that might be similar but slightly different, or they might be radically different, right, depending upon the final mRNA. So here's an illustration of this. So the blue line over here is a chromatin fiber. So this is along some chromosome, it doesn't matter what chromosome it is, right? And along it, we have RNA Pol II. This is all taking place in the nucleus. We have RNA Pol II transcribing. Maybe it starts from the top and it moves down, right? And the red line coming out is the length of the primary RNA transcript that you would see at that point coming out of it. So as a result, as the farther down you go, right, as you follow the RNA Pol II complex is going down, the size of the RNA transcript that each one of these is carrying gets larger and larger. I know this is kind of weird because I don't know why. I always had a hard time imagining what I'm really looking at here. So I think some of you probably do as well. Because what I always want to reemphasize is if this is a gene starting from here, where my cursor is, and the end of the transcript would be down here, that's why by the time you get down here, the full RNA that's photocopied from it represents the full primary transcript. The important thing to keep in mind is it's not like big purple RNA Pol II complexes, line up and then one by one, wait for one to get all the way down to the bottom before the next one starts, right? In reality, they're all continuously lined up after each other going down. I think it's kind of like a buffet at a restaurant. Like, remember, picture the last time you went to a nice buffet. It's so sad, right? That's one of the casualties of the pandemic. We don't have buffets anymore. But anyhow, you line up at the start of the buffet where all the plates are, right? And everybody lines up and takes a plate, right? And if the person in front of you takes a plate and starts working down the bread rolls, the salad, the soup, the rice, don't load up on the rice. They always put the rice early because they want you to load up on that, right? And then the good stuff is at the end. It's not like you stand there waiting for them to get all the way down to the end of the buffet before you start, right? No, it's like a whole line of people. And the point is, if you look at your plates, right, your plate from a person at the beginning of the buffet would seem very empty, not much on there except for a dinner roll, right, compared to the person at the end who has loaded up, right, on prime rib and stuff like that, right? So that's what we're looking at here, right? So as you're looking at RNA Pol IIs moving down this strand here, they're all carrying their RNAs that they're building. And towards the end, the ones that have gotten to the end, right, the RNA that's coming out is much bigger. It's probably funny is actually, I bet you I'm the only one who's confused about that. I bet all of you are like, no, I totally get it, Dr. Shen. You didn't have to come up with a weird buffet example, but that's okay because I always think about food examples. All right. So here's an electron micrograph of this. So this is cool, right? Because this is real. This is exactly what you can see under electron microscope, right? So the chromatin strand and the length of primary RNA transcript coming out. But then you look at these and you're like, wait, how come they're like bumpy and lumpy? Right? What's that? It looks like there's stuff attached to it. What's that stuff? That's the splicing machinery, right? So those are the proteins that have been recruited now to different introns and exons forming these lariats, right? So this is a region of, let's say, an intron that's going to get cut out of this particular RNA, right? So this pre-RNA comes down here and it kind of loops around and then it ends up over here. But in reality, all of this maybe is an intron that's going to get edited out. So the final mRNA would come down this way and just go off to the right here and end, right? So because of this, different RNA binding proteins binding to different spots along the transcript, like these black dots here, that's what they are, will influence, right, how different introns get cut out and how certain exons get kept. So in this case here, depending upon how these little black dots are binding, right, these are proteins binding to the nucleic acid, to the RNA, right, those will maybe block other proteins from getting in there and doing splicing, or maybe they will encourage other proteins to come and splice in those sites, right? So depending upon where these RNA binding proteins bind, that's how you get all these alternative products, all from the same starting gene, right? And to date now, I think there was a recent study that was published that shows that on average of the 20,000 genes that we have in our body, actually that number is kind of updated now. I think we're more like 25,000 genes that make up the human body. We make far more proteins, from that than you would guess if every gene only made one protein, right? So they've determined that on average, each gene, each protein coding gene in the human genome, on average, makes 3 different proteins from it. So that means some genes only make one protein from it. Other genes might be crazy and make 10 different proteins from it. But on average, it's like 3 different proteins per gene. All right. And it's all a function of this type of RNA editing. All right. So that what we've been talking about has been taking place in the nucleus, right? So in this cartoon here, the blue part, upper part is all in the nucleus. So this is a nice reminder, right, that transcription and the production of mRNA happens in the nucleus, along with, of course, right, the production of small ribosomal RNAs and the production of tRNAs, right? So look, here are the 3 RNAs that we've studied in the class, right? And this is a reminder that they're all made because they're transcribed from the DNA. Then the rRNAs, the tRNAs, and the finished edited mRNAs are moved out into the cytoplasm or the cytosol, where, of course, different proteins will come together to make the ribosome. Right? And here's the mRNA that was moved out of the nucleus, right? So the ribosome can go to work now on translating that RNA. The ribosome is made from proteins as well as the ribosomal RNAs that were made up here. And of course, the tRNAs are helping out to bring the amino acids together to make then the finished protein product of what this gene was meant to code for, right? So the cool part about this figure is this second part that we don't really think about, right? A lot of these proteins are destined to do their job back in the nucleus, because after all, these are proteins that are, for example, the histone proteins, right? The histone proteins that make up the nucleosomes. They had to have originally been made from the gene that codes for the histone proteins, right? And they went through this process of being transcribed and translated only to have the finished histone protein go back and do its job, controlling whether the chromatin will allow other genes to be transcribed, right? It's like this amazing loop in a way, right? And I think that's really cool to think about. After all, the ribosomal subunit proteins themselves came from this process, right? And even the RNA polymerase II complex, the big purple complex with the tail that does the transcribing in the first place, all of its subunits are proteins that were coded for by genes as well, right? And you're like, where does it end? Where does it start, right? Because you can't have this process happen without those proteins, and you can't have those proteins without this process having started, right? That's the amazing thing about life, right? And that's all part of how you went from 1 cell to 30 trillion cells. Okay, so let's finish up talking about some other examples of how the RNA itself, the mRNA itself, as it's getting ready to be translated by the ribosome, itself can be a target of the cell, right? As it's sort of another way of controlling whether protein even gets made from it. Okay. So ribosomes are riding along the mRNA to translate it, right? So you can imagine that other proteins that bind to the mRNA could actually interfere with this process. So this would be another way that the cell can control whether protein even finally gets made from the mRNA. So this is kind of similar to what we learned last time. We said certain proteins can bind to DNA, right, and act as repressors to block things like RNA Pol II from coming to the promoter and even transcribing the gene in the first place, right? Now we're talking about, well, if transcription happened and you actually have an edited mRNA, right, it's also this, genetic molecule, right? Instead of DNA, it's RNA, but hey, proteins can bind to it and then block the next level of the process, block the ribosomes from doing the translation. So here's an example that helps to highlight this. Iron, of course, is a very important component of our tissues. Iron is used to make things like hemoglobin, for example, that our red blood cells carry around. Right? So iron has to be transported through our blood. it's usually transported, not just free iron, ions floating around in the blood. It's bound up by proteins called transferrin. So the transferrin, you can see here, carries a couple of iron ions on it. So how is that going to get in the cell? Well, a cell that needs to use the iron would need to have proteins on its surface that can bind to this transferrin and get the protein in, get the iron in. So this Y-shaped thing is the transferrin receptor protein. So it's on the surface, it binds to the transferrin that's going through the blood, and then it can take the transferrin into the cell where the iron can then be removed, right? And the iron can then be used to make things like hemoglobin, right? If you have extra iron, let's say, the cell made plenty of hemoglobin, it doesn't need to use more iron. The cell doesn't want to just lose it because that's, valuable stuff. Maybe extra iron can be bound up to this other protein known as ferritin. Okay, so transferrin receptor is the thing that helps bring the iron in when your cell needs it. The ferritin is a protein that can store the extra iron, right? So that if you need it again, it can be released. So how might varying iron levels control the translation of these proteins? So here we have an example where if you have a lot of iron in your blood, right, you would want to make a lot of the ferritin protein that can store the extra iron. Right? But if you don't have a lot of iron, right, then you don't need to bother making that storage protein. Right? So here's the ferritin mRNA. Right? So the ferritin mRNA is now in the cytoplasm. Your ribosomes might go ahead and translate this into the ferritin protein, but in this case, there's no iron around, so your cell doesn't want to waste its time and resources doing that. So along the mRNA, there's a sequence known as an iron response element. So this is kind of a generic way of just saying that somewhere in here is a sequence that certain proteins recognize and are sensitive to varying levels of iron. So here, this dark purple thing is an IRB binding protein. It recognizes and binds to it, and it blocks the ribosome from being able to come and translate this mRNA. However, if you have a lot of iron in the cell, and the cell now needs to make this ferritin to store that extra iron, right, that ferritin binding protein, or the inactive IRE binding protein, binds to the iron, and that removes it from the mRNA. So now the ribosomes can start translating, right, and making the ferritin protein that can now help by binding and storing this extra iron. The opposite would be something like the ferritin receptor. Right? The ferritin receptor, if your cell doesn't have enough iron in it, then you want to have a lot of this ferritin receptor protein made to put on its surface to be able to catch iron or the transferrin that's coming to the outside of the cell and bring it in. So that means normally in low iron conditions, the transferrin receptors mRNA, right? This is a different mRNA than the one we saw on the previous slide. That was the receptor RNA. That was the ferritin mRNA. This is the transferrin receptor mRNA. Normally, in low iron conditions, the ribosomes are busy at work making plenty of the transferrin receptor protein. Right? There is also an iron response element sequence in this mRNA, right, that has this particular IRE binding protein bound to it. And what that does is this protein protects the mRNA from being degraded by these blue triangle enzymes that would love to come and cut up this mRNA. Right? However, if there's plenty of iron around, right, then the cell no longer needs to make this receptor on its surface anymore. Right? And as a result, the iron binds to this IRE binding protein, and in a similar sort of theme like the previous slide, it removes that IRE binding protein from the mRNA. And as a result, what happens? Now these degradative enzymes can come in and chop up the mRNA, destroying it, right? So your cell has decided to stop making finished protein from this mRNA. It's happy just chewing it up and degrading it. Okay, so I have a couple more slides, which I think Well, I can get to those of you who have to go. You can go because you can always watch this on the YouTube. But I want to give you a cool example that I think you will like. So if you can stick around, listen to this. Right, there is a mechanism inside of our cells known as RNA interference, and it uses this principle of if you want to stop an mRNA from being translated into a finished protein, one of the best things you can do is simply just chop up the mRNA before ribosomes have an opportunity to translate it, right? And you say, why would I want to do that? Remember, right, free-floating mRNA in the cell cytoplasm, our cells evolved to be kind of worried about that, right? Because free-floating RNA that has no other sort of sequences in it, like a cap or a polyadenylation tail, it could be from a virus that just infected the cell. Right? So our cells want to destroy these mRNAs. But you might say, well, you don't want the cell just to like go crazy and just destroy any mRNAs that are coming out of the nucleus, because, I mean, obviously, a lot of the mRNAs normally are the ones that your cell are making on purpose, right, to be translated into proteins. So there's this evolutionary mechanism known as RNAi. And the way it works is this. Here we have some double-stranded RNA. Let's say it comes from a virus, right? So it's kind of weird because it's double-stranded to start with, right? Because our mRNA is not double-stranded, right? So right away, our cells evolved, hey, this is not right, right? We need to get rid of this. So you have a protein complex known as dicer that dices up the RNA into small little pieces, right? So, okay, great. So now you've destroyed the viral RNA. It won't now be a threat, right? You could just stop there, but our cells do something even more remarkable. There's another complex known as a RISC complex, this yellow blob here. This RISC complex targets single-stranded RNA for degradation. Single-stranded RNA like the mRNA that comes out of our nucleus, right? But then you say, wait, but I thought the whole point was we wouldn't want that because then we'd destroy our own mRNA. You'd have to be able to distinguish between our mRNA and viral mRNA, right? Because let's face it, if the virus infected this cell, it's not like there's only this one double-stranded RNA that went in there, right? Chances are there's already the viral genome in our cells, and the viral genome may already be hijacking the cell to create viral mRNAs, right? So what this RISC complex does is super cool. It actually takes one of these little pieces of chopped up RNA that the dicer made when it destroyed this example of the virus, and it loads it up as a template. Right? Now, when it encounters mRNA that's coming out of the nucleus, and that mRNA gets kind of scanned by this RISC complex, if any part of that RNA sequence matches the little guide RNA template fragment that it's carrying, then the RISC knows this is not our RNA. This matches the sequence of the viral RNA, right? And it will go ahead and preferentially destroy that viral mRNA before our cells accidentally turn it into new virus particles, right? So I think that's really cool, but it gets even cooler. Some of you, if you're still here, might say, wait a minute, right? This means there's the potential to actually guide this RISC complex in our cells to destroy certain mRNAs coming out of the nucleus that we want it to destroy, right? I mean, evolutionarily, Mother Nature intended this to be, we recognize viral RNAs coming out of the nucleus. But what if you actually have people who are born with maybe some polymorphism, some defect in their genes, such that the mRNA that's coming out from that gene from the nucleus into the cytoplasm is actually defective in a way that if it was translated into a protein, right, you end up with some harmful disease-causing version of the protein. What if you could actually target and destroy that wrong mRNA as it comes out of the nucleus before it ever gets turned into a protein to cause harm? And so that's what we've done now, right? So this is really pretty new technology. There was a company, European company, who took this concept and designed the first RNAi drug as a therapeutic agent. So it's called patisiran. The market name is Onpattro, right? And it was approved by the FDA back in 2018. This is the first time there's ever been a interfering RNA based drug. What they did was they started with a particular disease. There's a type of hereditary liver disease that causes a defective version of a liver protein to be made in hepatocytes, liver cells. So people who have this disease, they inherited an allele where one base is different in this gene for this particular protein, and that results in a different amino acid being at that particular position in the finished protein. And that one amino acid change causes the protein to fold in such a way that it precipitates out of solution and it forms solid plaques, right, of this misfolded protein that ends up doing things like damaging the tissue and can actually be fatal as it interferes with things like nerves. Right? So this protein is the transthyretin protein, right? And it causes this fatal amyloid plaque buildup in the livers, right? And so the researchers, they designed a small fragment of RNA that has, on purpose, this one wrong DNA base in it, right, that the people with this disease would have as their genes, right? And they've packaged this small interfering RNA in a little lipid bubble so that it could then be delivered to the cells. And what it does now is when this enters the cells, right, the dicer and RISC complex recognizes this RNA as being something foreign, right? It loads it up in the RISC complex, and now when these people's livers are making their defective version of the mRNA from their defective gene, right? Instead of the ribosomes having a chance to translate that and make the fatal protein, right? Their cells now recognize it and destroy it. Right? So I think that's amazing, right? And you know, it's something, like I said, this is the first example of using this technology to be able to actually leverage your own cells to do what they already know how to do, right? Which is to chop up unwanted mRNA. But now you're teaching them that this particular mRNA that's coming from the nucleus is not wanted. And so they should chop it up. And it works, right? Because now these people have hope for actually surviving this disease. Okay, I'm sorry that I went over today, but I wanted to put that in there because hopefully, you know, this helps you really kind of appreciate what we're talking about when we're talking about our cells being able to target mRNA as a way of controlling gene expression and the production of proteins. Okay, let me stop the share.